I'm going to talk about the basics of this cell model. It's not showing all the structures of a typical cell model. For instance, it's not showing lysosomes or peroxisomes, but it's got the basics. So here is the nucleus. The nucleus is where we contain our genetic material. So our DNA is in the nucleus, and we will copy our DNA in the nucleus, and we will make RNA from DNA in the nucleus, and then the RNA can exit. It can exit out a nuclear pore. So this is the nuclear envelope. It's the fancy word for nuclear membrane, and it has nuclear pores on it. You have chromatin, the very diffuse extended form of chromosomes. And so when we are not dividing in interphase, our genetic material is in the form of chromatin. It will condense into the form of a chromosome before we divide the nucleus. Then the membrane of the nucleus flows and becomes the membrane of the rough ER. It's called rough ER because it's studded with ribosomes, so it's going to make proteins that exit the cell. We have the smooth ER. It is not studded with ribosomes. So the membrane of the rough ER will become the membrane of the smooth ER, and the smooth ER will make lipids, store calcium, and detoxify drugs. Here you can see several mitochondria. The mitochondria are the powerhouses of the cell. They are going to make ATP, utilizing cellular respiration. You have ribosomes in the cytoplasm. Ribosomes in the cytoplasm make proteins that stay in the cell. Ribosomes on the rough ER make proteins that exit the cell. Here you have the Golgi apparatus. The Golgi is going to process, store, and ship the products of the ER. So you have a cis side, which vesicles will come in and fuse, and then they will be transformed. Likely they'll have like a targeting signal put on them, and then they will come out the trans side. Let's see. So nucleus, nuclear envelope, nuclear pore, chromatin. Inside the nucleus, we have the nucleolus, which makes ribosomal RNA. Ribosomal RNA will exit the nucleus and form ribosomes, and ribosomes are where we make proteins. If the ribosome is on the rough ER, it makes proteins that leave the cell. If the ribosomes are free in the cytoplasm, it makes proteins that stay in the cell. Mitochondrion makes ATP. Smooth ER stores calcium, makes lipids, detoxifies drugs. And Golgi processes, stores, ships products of ER. And we have the centrosome, which in an animal cell has these two structures called centrioles at a right angle to each other. The centrosome is the microtubule organizing center. So you'll have, for instance, during mitosis, the spindle apparatus, which has microtubule fibers radiating from the centrosome, will be formed. And so, the cytoplasm is, the inner material of the cell, and then we have our cell membrane. So that is the basics of this cell model. Keep in mind it doesn't have all the organelles listed, but that's a discussion of this model.